Simon Frost is an AHC parent to Annabel. For those of us who don't know, he's a husband to Nina and a father to Clara, Annabel's older sister. Like all of us, Simon has been thrust into a world he probably never expected. He and his wife have had to become pseudo-experts, like many of us, in a rare disease, in genetics, in pharmacology, and in patient care, and in balancing an additional career's worth of work to seek out a cure for AHC. Simon, in his normal life, is the CEO of Tiber Capital Group. He served as the executive team of several large finance and real estate companies. Most recently, he was the chief investment officer of Green Court Capital, a public company with approximately $1 billion in real estate assets. Simon was the co-founder of the American Home, one of the largest single-family rental aggregators in the United States, and was the president and chief operating officer of Key Properties, another large single-family rental aggregator. Simon has acquired, renovated, leased, and sold over 10,000 homes since 2011. Simon holds a bachelor's and master's degrees in economics from Cambridge University in England and a bachelor's degree in finance from the University of South Africa. Simon serves as director of both Cure AHC and Hope For Annabel. And those are charities dedicated to finding therapies for our disease. He's played professional rugby in the UK for Swansea, London Welsh, and Coventry, and captained the Cambridge University's rugby team. In short, Simon is a pretty bright human with a hard head. Simon has applied much of his training to take the lead in genetic therapy effort in AHC by engaging with scientists and researchers, developing a strategy to transgene therapy in AHC. Today, Simon will discuss the status of the Transgene Project. This is the project that's been referred to as the AAV Project, a project brought to Cure AHC by Simon, subsequently brought to the AHC Foundation in 2018. This is a project that has formed a strong collaboration of family organizations over the last 3 years. Through Simon and his collaborators and our team, I think I've personally earned a master's degree in biology. I know he will elevate your understanding of both AHC and the leading edge of gene research. Please welcome Simon Frost.
Thank you, Josh.
Josh, can you please also tell people how to get to the sibling room and let Jean introduce Ryan, because that's also going on right now.
Thanks for the reminder. That's right. There is a sibling discussion hosted by Jean's son, Ryan. And after Jean introduces Ryan, if the siblings would like to go down to the breakout room in the bottom of your window and click join next to the sibling room, not the kids' playroom. But please do that after Jean introduces Ryan.
Thanks, Josh. So Ryan is Kelly and my almost 22-year-old son. He's studying electrical engineering with a minor in business at the University of Wisconsin-Madison, and he'll be graduating this December. He's currently working as an intern at Collins Aerospace in Cedar Rapids, Iowa. As Kylie's older brother, he's seen the effects of AHC physically with episodes and seizures, developmentally and emotionally and behaviorally. He's often a calming influence on Kylie and someone she looks up to and respects. He has 20 years experience as a sibling and has been a peer minister at the St. Francis de Sales Church, and he's led AHC sibling sessions at previous family meetings. The session he's leading today is for all AHC siblings to share their feelings, questions, and ideas about their experiences as an AHC sibling. As a reminder to parents, the sibling session is for the children only, and it's a time for them to express their feelings freely. That session will not be recorded. So Bill, if you can open up that room and the siblings can go and have hopefully a very meaningful discussion.
Yep, it should be opened on the bottom of your screen. If you hit breakout rooms, you should be able to switch over to sibling meeting Ryan.
All right, can everyone see my screen? Yep, Good to go. Okay, so thanks so much for inviting me to provide an update on AHC therapy research. I'll start quickly with a history of our collaborative efforts, and then I'll talk about where we're going on transgene delivery, which is what we call our AAV project. Then gene editing, which you just heard about so eloquently from Alex Souza, and then other options we're pursuing like drug screening, antisense oligonucleotides, which are also known as ASOs, and RNA options like shRNAs, siRNAs, and other knockdown tools that might combat potential dominant negative effects of specific gene mutations. This has been a widely cooperative effort involving all the foundations and more than 50 leading neurologists and scientists from many hospitals, labs, and research institutions around the world. I call this a parent-led initiative, and it is, but it's much more than that. It's a network of the top minds in our disease working together in a uniquely collaborative way, not for researchers' personal resumes or for grants and funding, but truly to help our children. This collaboration kicked off in September 2017, when, as a team of patient advocates and advisors, we jointly started looking closely at AHC with a stubborn focus on advancing therapeutic development. We were tired of uncoordinated science driving us and decided instead to drive the science by directing research efforts. We now decide jointly which research to pursue to speed the path to treatments. It's been a powerful consortium that has become a shining example in the rare disease world of how parent-led research can significantly influence the future of rare diseases. We started by looking closely at the disease, the gene that causes the disease, and many hundreds of research papers on wide-ranging topics. We started with the clinical diagnostic criteria and disease manifestations. Then we looked very closely at the molecular and cellular basis for the disease. We organized the research into logical areas: molecular structure, function, clinical studies, phenotyping, genotype-phenotype correlations, electrophysiology, in vivo and in vitro experiments, treatment research, drugs, gene therapies, mouse studies. We got together as a community and we devised a research plan to target potential therapies. As a parent-led initiative, we acted as a hub to consolidate all of these different topics. We laid out all possible therapies, but there were essentially only two that were viable: drugs and genetic therapies. There were two categories of drugs: on-target and off-target drugs, and four categories of genetic therapies: transgene delivery, antisense oligonucleotides, other knockdown options, and gene editing. We looked closely at the molecular dynamics of the sodium-potassium pump and the molecular basis for the disease. The sodium-potassium pump is a very active protein with no obvious targets for binding of target small molecules or drugs that might help the pump to function or stabilize dysfunction. The more the team researched, the more we started to see some interesting neurological patterns in AHC that we could target. So as you know, the ATP103 gene codes for the sodium-potassium pump, which is supposed to pump sodium ions out of neurons and potassium ions in at a ratio of 3:2 to create and maintain the electrical and the chemical gradients that are required for neurons to fire and keep firing. ATP103 pumps work particularly fast compared to ATP1A1 pumps, and when they fail, as Dr. George so clearly showed us yesterday, intracellular sodium ions and extracellular potassium ions build up, resulting in depolarization and neurons that stop firing. We noticed that Gabaergic neurons, which are inhibitory neurons, fire at much higher frequencies than glutamatergic neurons, which are excitatory neurons. Those fire at much lower frequencies. It seemed that high frequency neurons that have long firing cycles would probably fail faster than low frequency neurons if they didn't have functional pumps. So we looked closely at those high frequency GABAergic neurons. we located the highest frequency neurons. We correlated those locations with known atrophy in the brains of AHC patients, and we saw that there were clear patterns in certain regions of the cerebellum, basal ganglia, hippocampus, and frontal cortex. Then we looked at ATP1A3 expression by cell type, and we found that almost all neurons co-express two different types of sodium potassium pumps, ATP1A3 and ATP1A1 pumps. But a particular type of Gabaergic neuron in the cerebellum called Purkinje cells only express ATP1A3. Those cells don't have a safety net, and when they fail, there's no backup. We also learned that these Purkinje neurons have gap junctions which connect to other cells directly. These gap junctions allow sodium ions to diffuse across them. So when one Purkinje cell fires frequently, depolarizes, and fails, those concentrated sodium ions can spread by diffusion to neighboring neurons, causing them to depolarize and fail. And what results is something called spreading depolarization, which can be damaging to the brain, and of course is terrifying. So that became our thesis, and we traveled around the world refining it, challenging it, and testing it. We met with scientists from Aarhus University, where they won the Nobel Prize for identifying the sodium potassium pump, to Columbia and Duke, where they found the disease-associated gene, the NIH, which has now established a dedicated lab, and conferences around the world. If we can stop those Purkinje cells and other high-frequency parvalbumin-positive GABAergic neurons from failing, then perhaps we could prevent spreading depolarization, brain atrophy, and those so-called human time bombs that we all live with. Then we focused our research. We looked at drug and small molecule screening. It's not precise research, and it relies on a bit of luck to find a new molecule or a repurposed drug. But it's been a technique that's been used successfully in other diseases. As you may have seen, the European AHC foundations have now secured a grant of almost 400,000 euros for a 3-year study aimed at identifying candidate compounds to treat AHC and testing those compounds in vitro and in animal models. This will hopefully culminate in a clinical trial, but there's a long way to go. At the same time, we were also looking at drug options, we were also looking closely at gene therapies, which has been a tool that's been used successfully for over 20 years in cases like our AAV-mediated gene therapy to treat rare diseases like ours, and most of the research we funded so far has been in this area. In 2018, we kicked off our gene therapy work with the AAV project. The objective was to deliver as many functional copies of ATP1A3 genes to affected neurons as possible, especially to Purkinje cells. We developed mouse colonies, cell lines, viral vectors, experimental protocols, and behavioral studies. AAV-mediated gene therapy involves packaging the gene, in our case ATP1A3, into a benign virus and delivering it in large quantities to the cells that need it. We chose the specific sequences of DNA that we wanted to deliver, the manufacturing processes we wanted to use, and the delivery sites we thought would work best for AHC. As you can see here, we thought the cavity at the top of the spinal cord, called the cisterna magna, would be a good delivery site for a disease such as ours with so many cerebellar symptoms. We asked some of our friends if we could use their mouse models. Most said yes. One critical friend said no. So we decided to rederive that mouse ourselves. That rederived mouse recapitulated the human condition very well, with obvious targets for rescue. It's important to know that very few mouse models replicate the human condition so well, so we got lucky. You can see an example in the chart on the left that shows that every single AHC mouse gets dystonia when you put it into cold water, and every single mouse that doesn't have AHC does not. If we could somehow show that some of our mice were not getting dystonia after being treated, it would be a glaring example that our therapy was working. We started our AAV project by using IVF to expand our colony rapidly to over 200 mice. We injected over half of them on the day they were born with either an empty capsid, and that's treatment group 1 in this slide, or a therapeutic vector, which is treatment group 2. We did genetic screening after the mice were born, and unfortunately, because mosaic sperm was used for the IVF expansion, we got a low number of AHC mice and a large number of pups without AHC, which was our first setback. Even so, early body weight data suggested that we might be onto something. You can see in the chart on the right that by 4 weeks of age, our treated AHC mice were significantly heavier than untreated AHC mice. We soon suffered our second setback. Sadly, during a move from the storage location to the behavioral testing suite, many of our AHC mice died, presumably because of cage rattling, bright lights, and noise. As you can see in the charts here, the survival rate plummeted. Many of our heaviest mice died, and body weight data as a consequence no longer looked promising. These unfortunate events in our first pilot meant we needed to recalibrate for our second pilot. We redesigned the study to make sure the mice weren't moved on shaky trolleys. We also worked to identify other potential causes of death in our mice. We looked at the biodistribution and expression of our vectors across the mouse brain to see if we could do anything differently. Jackson Laboratory, which was running our trials, did a ddPCR analysis to see whether new DNA was showing up in the right places in the injected mice. They found a very high number of ATP1A3 copies in the front of the brain, but a low number in the cerebellum, which was not ideal. We'd injected the vector into the center of the brain, which had proved to be suboptimal. So we changed the delivery site back to the cisterna magna. We looked at our vectors for potential toxicity. We looked at the DNA sequence, including the promoter, to see if we could optimize where the vectors were expressing. We needed the vector to express in the right parts of the brain and showed no expression in the wrong parts. We also looked at dosage testing to make sure we weren't overdosing the mice. We decided to make a few changes in our second pilot study. We changed the promoter to make our vector express only in neurons. And in the slide, you can see the changes we made in the colors. We changed the method of production to use mammalian cells instead of insect cells. As I mentioned, we decided to inject one cohort of mice into the center of the brain, which counterintuitively is quite easy to do, and another cohort into the cisterna magna, which is quite hard to do. We had a lot of mice at this point, so we decided to use a breeding colony instead of surrogate mothers. And unfortunately, this was also problematic. The surrogate mothers we used in our first pilot were a strain that had been chosen for their motherly instincts, and it turns out that the mothers in our model, the C57 Black 6 strain, are terrible. We had a big problem with cannibalism, and we lost a lot of pups. I mentioned that injecting into the cisterna magna is really hard to do. The techs at the Jackson Laboratory are skilled, but they needed to show real proficiency before we kicked off the second pilot study. We would only start the third pilot once they could demonstrate clearly that they were hitting the target at least 90% of the time. And in this slide, you can see they hit the target 12 out of 12 times with blue dye. Even though we'd lost a lot of pups through cannibalism, we still had enough injected pups to run tests in pilot 2. Our cohort had been injected into the center of the brain on the day they were born, which is the chart on the left. And the other cohort had been injected when they were about 4 weeks old, which is the chart on the right. You can see both of these charts show some real promise. The AHC pups injected both ways have showed much better survivability rates for treated pups versus untreated pups. You can see in the chart on the right that all the pups that were injected into the cisterna magna are still alive after more than 100 days. You can see that again here, which is a chart that compares pilot study 1 to pilot study 2. When we compare the survival rates covering both the injection sites to the natural history data and to the data from the first pilot for post-wean mice only, mice that had got through the first few weeks of life and had been weaned, the difference is quite stark. The survival rate is 100% compared to much lower numbers for pilot 1, and compared to the natural history data for the typical phenotype. We're also seeing some compelling data from hypothermia-induced dystonia testing. In pilot 1, as I showed you with the mouse phenotype, 100% of AHC mice got dystonia, and 100% of mice without AHC did not. In pilot 2, it was a very different story. Several treated AHC mice did not get dystonia. Recovery time is also looking promising. Recovery time appears to have decreased in pilot 2, and this is the recovery time after being in cold water. Note that even wild-type mice, so mice without a disease, require some recovery time before they start grooming and doing typical mouse things. So the recovery time will never be 0, but there does seem to be a significant decrease. Not statistically significant because we have a very low population of mice, but from the charts, it looks like there's a decrease in the recovery time from pilot 1 to pilot 2. So these data are very promising. We're excited to step into pilot 3 with the same vector we used in pilot 2. You can see that the changes here are positive changes involving the surrogate mothers and the heterozygous sperm. We're adding one test, which is going to be the Erasmus ladder to test for ataxia, which is also a statistically significant test that we've found from other papers. So we will be using a nice clean vector in pilot 3 with no tags, no fluorescence, the same type of clean vector that we might be able to use in our children. As you probably know, a mouse model has now been rescued independently using these methods that we've produced and patented and continue to protect. This rescue is established proof of concept for AAV-mediated gene therapy for our disease. It looks like this therapy is now on a path through the FDA approval process with the goal of reaching clinical trial. We're working hard to deliver a better, more robust rescue using only the delivery into the cisterna magna and a far cleaner vector than was used in the rescue paper. Either way, the rescue paper is exciting proof that our patented approach works. As you just heard from Alex Souza, we have started research on gene editing, which is cutting edge science. We've signed a 2-year contract with David Liu and Alex Souza's lab at Harvard and the Broad Institute to investigate base editing and primate editing in AHC. This study will culminate, as we said, in an in vivo proof of concept behavioral study in our mouse colony. I'm sure you can now see that gene editing is not some distant thing. It's real, it's happening now. You saw that Dr. Liu's lab has rescued a mouse model of progeria. using base editing. And they've shown for Niemann-Pick type C that very high transduction and editing ratios can be achieved in Purkinje cells, the very cells we're targeting. And as you just heard, the initial data has come back positive for prime editing, and we're pushing hard towards proof of concept there too. We're also investigating RNA knockdown options to target the mutant allele and antisense oligonucleotides to preferentially moderate the expression of the bad gene and potentially upregulate the expression of the wild type allele or other background genes. In other words, we're looking for ways to improve the life of patients using every genetic tool available to us. The good news is that we can theoretically use these therapies together. They can be complementary. We can theoretically compound them by delivering transgenes, rescuing with gene editing, moderating using antisense oligonucleotides, and knock down the bad copy of the gene with RNA tools, all in the same patient. It's of course not easy or immediately realistic, but it's one of the reasons we're pursuing all of these therapies in parallel. So we're working hard to create repositories of virus-mediated gene therapies, antisense oligonucleotides, other knockdown options, and gene editing machinery. We're coordinating and to centralize genome data, deep phenotype data, natural history data, and apply those big data analytics to target therapeutic research. And this doesn't stop with AHC. We're actively advocating to centralize mouse models and cell lines and test all 9,500 rare diseases using plug-and-play therapies like AAV vectors. We're collaborating with big institutions like the NIH and RareX to create replicable protocols for other diseases to follow, to target research based on producing therapies quickly for their diseases and for their children. With over 300 million people affected by rare diseases, it's one of humanity's biggest challenges, as you all know so intimately, living with AHC. Thank you.
Thank you, Simon. There's a question in the chat from Dr. George. At what age are the mice treated by both ICV and ICM routes?
So ICV is P0. So as soon as they're born, they're injected ICV. And ICM is day 28 or thereabouts. It ranges between 25 and 35 days.
The only question we have in the chat. Does anybody in the room have questions for Simon? Simon, you did a great job. You always do a great job bringing this kind of stuff to family level. So I appreciate it.
Thank you, Josh.
Oh, here's a couple of questions coming in. Meredith asks, has trial 3 started yet?
We have pulled the trigger on the IVF expansion, and that's going ahead. We're waiting for a date, and that date needs to be coordinated with the Erasmus ladder testing. So we're trying to figure that out. But it takes 4 weeks for mice to be born after the IVF expansion. And at that point, they'll start being injected ICV.
Sveta asks, is this research going to be published?
Yes. So the whole kind of point of the exercise is to get to proof of concept. We need statistically significant data to be able to prove the concept. But yes, we will be writing as fast as we can once we do have that proof of concept. And by we, I mean our scientists, our PIs.
That leads right into another question. Can you remind us who the we is in this work? Who are the researchers doing the work?
I really should have put a slide together on this, but there are more than 50 people that are helping, and I can't kind of name all names, but I'll start with the foundations. I mean, AHCF has been a huge pioneer here. We've partnered with Hope For Annabel and Cure AHC, and all foundations are kind of helping each other to make decisions on this and really poking holes in the thesis and poking holes in research grants and making sure we're making the right decisions. So that's been a big part of it. We also have very highly qualified neurologists and scientists. The best that we have really are all on board, bar maybe one or two. And I mean, I could go down the list, but everybody on this call is hugely is participating hugely. Dr. Swoboda is a great example. Dr. S, Dr. George, but there are so many others. Dr. Klapko, Dr. Licky Hartman. We've got scientists in basically every area that's are top of their field and know minute details about how to handle mouse models, how to inject. It's been a huge collaborative effort that I can't, describe in a very short few sentences.
All of these therapies are parallel. Is one favorable over another once FDA approved?
I guess it's going to be the one that works best. It's very hard to say which one that's going to be. I have very high hopes for AAV mediated gene therapy. It goes all the way back to a paper by Dr. Klapko where he added a lot of genes by a method that can't be used in humans to a lot of ATP1A3 functional copies and rescued a mouse model that way. And we tried to replicate that with AAV mediated gene therapy. And it looks like the data would suggest that we can get there on that. It's something that has now been proved. The concept has been proved, but I would like to see a more robust rescue before I kind of inject my own daughter and would recommend that with everybody else. But I think we can make this work better, and we can do that in a short time frame. Gene editing is obviously a huge opportunity for us. I don't want to kind of overstate what that opportunity is because it would be unfair to the patient group, but it's exciting. It's new. It's cutting edge science, and it works, as Alex showed us so clearly.
Thanks. We have a question from Catherine Bell. You mentioned around 2+ years time frame for clinical trials if things go to plan. Can you explain what that will look like? Will it be USA based? Who will be overseeing the clinical part in terms of clinicians, and what age group will you start with? You have previously mentioned starting with older AHC children due to ethics.
Yes, so I'll start answering that question by saying that I think that that there's a kind of predetermined path for every disease to get through approvals and to get through the manufacturing process. So just like most viral deliveries of therapies, you need to go through a toxicology stage, which Josh, you described yesterday, and I think Jean described yesterday. That takes a little while. You normally do that in rats. It costs a bit of money to get it done, and then you need to manufacture the therapy at scale, and it requires huge manufacturing facilities to produce the amount of viral vector that you need for delivery into humans. Then you need to design and execute your clinical trial, decide who the clinician is going to be, where it's going to be, which hospital it's going to be in. You have to decide how many patients are going to participate, what the qualifying factors are. And, we will be working as a group of foundations to make sure that we make the right decisions on qualification factors. But in other diseases, patients have qualified regardless of their genotype. So just because we're testing things on D801N for the delivery of a transgene, right? And this is not gene editing, obviously, but the delivery of a transgene is delivering a functional copy of ATP1A3 that should dilute the effects of the dysfunctional copy of the gene. That can be proven in cells to qualify patients, and you don't necessarily need to have a D801N mutation to participate in a clinical trial. The age of patients, I think that was just my opinion, Catherine, that I would prefer to see older patients do it than younger patients. I think that tends to happen in gene therapy trials, which actually skews the data a little bit, because the earlier you can catch, that you can kind of counteract the effects of the disease, the better. So the data gets skewed kind of not suboptimally. But those are all factors to consider going forward, and that's kind of why it takes 2.5 years plus to get there.
So the next question is from Thomas O'Brien. What is your confidence that we will find something? And 2, is there a risk as we focus on one mutation, other mutations may be left behind? Maybe we have no choice. How quick could we affect a treatment on other mutations if it works on the first?
Yeah, so confidence level, I mean, it's hard not to say 100% because we have proven the concept, right? It's, I think, that's been the point of the exercise up to this point, is to prove that a AAV-mediated gene therapy can effect a statistically significant rescue. So that has happened, right? It's not really a guess as to whether or not this can be an effective treatment option. I think it is now an effective treatment option. Now it's about how do we deliver it in quantities large enough to help our kids, and how can we do that in a safe and effective way? So, can this be an option for us? I think the answer is yes. We've got lots more work to do before we can inject it into kids, but I think we've got, promising is probably not a strong enough word, but good therapies that are now available to us. And I've failed to address the last half of that question. Do you want to ask it again, Josh?
Sure. Is there a risk as we focus on one mutations that others will be left behind? How quick could we affect a treatment on other mutations if this one works? And just another point, he wants to avoid a hierarchy of mutations within AHC.
Yeah, and that's a really good point. So just to be clear, my child does not have a D801N mutation. She's got an L839P mutation, which is, there are only 2 other known patients in the world with that mutation. Tom, I kind of feel exactly what you feel, because, the question is my child's mutation getting enough attention?
Mm-hmm.
And the way that I think about it is, if we figure out, that gene editing works for D801N, or that transgene delivery works for D801N, well, guess where I'm piling all my research money and attention, right? Into that particular path for L839P. So the fact that we're doing this in D801N doesn't mean that it doesn't help other patients. It means that it gives us more and more of a precise target to focus our research dollars and attention on for everybody. And it doesn't even extend only to ATP1A3 mutations. I mean, CACNA1A mutations, we have kids with those, with genetic mutations in that gene. We have other genes that are also affected, I think SCN1A. So if it works for our disease, we're not just, helping D801N kids, we're helping far more than that.
Yeah, I think just to add on to that, I think, Simon, we have discussed how the system that we're developing can be applied, or we hope could be applied to nearly 10,000 other monogenic diseases that don't even get any attention at the moment.
Exactly, yeah. So that's these are homogeneous therapies, right? Delivering a transgene to dilute the effect of a mutated gene is not something that's unique to AHC. It's something that can be used across many diseases, and there are 9,500 other rare diseases affecting 300 million people. It's there's a much bigger picture here, and the fact that we're on the cutting edge of science, especially with gene editing, means that we're helping more than our 1,000 patient community. I think we're helping a large number of monogenic diseases.
I think we've answered this, but I'll throw it out there anyway. Will AAV gene therapy be applicable for CAPOS patients or only for AHC patients?
So that's a great question. Dr. George is actually really qualified to have an opinion on this particular question. So if he wants to jump in, but we look at the electrophysiology of these different mutations. Some of them do appear to have more of a dominant negative effect than others. The CAPOS seems to be kind of almost opposite to E815K. They're both an E to, an E amino acid to a K amino acid substitution. And one's E815K, one's E818K. It's very interesting to kind of note that and look at it as to kind of understand those two mutations a little bit better, since they are both so severe. But the idea remains the same. We're diluting the effect of the mutant allele. And if we're doing that, then theoretically, if we throw enough kind of functional copies of the ATP1A3 gene, there would be dilutive effect as long as the dominant negative effect of that particular mutation is not severe enough.
Okay, we're right up against time, so I'm going to ask this additional question from Catherine. And I'm going to read it. In clinical research in AHC patients, there's a suggestion that cerebral atrophy occurs over time in some AHC patients, i.e. the MRIs previously were said to always being normal, but this is now less sure. Has this been studied in the mutant mice, and in particular in terms of the mice that are rescued with the gene therapy? If this alters cerebellar, I can't do that, volume as another objective outcome to effects of the gene rescue, as well as the outcome changes in effect in clinical symptoms in the mice, such as paralysis, dystonia, and ataxia?
Yeah, so that's a really great question. And now I'm going to kind of put all sorts of caveats behind my answer, because I'm obviously not a neurologist and I'm not a clinician and all that stuff. This is my opinion only, right? We see atrophy in patients, and we see atrophy in the brains of mice, and we see that a real cerebellar focus on that atrophy. This is kind of part of catching things early, right? And it's also got to do with the motor loop. So we know that this disease really affects the motor loop, and the thesis is that it starts in the cerebellum. We see a lot of motor related issues in our kids. It's very hard to say whether or not kind of transgene delivery or even gene editing would correct for the cerebellar dysfunction, and stop those paroxysmal events if there is an associated atrophy that's already happened, right? So I don't know the answer to that. What I do know is that we're doing our very best to kind of prevent that spreading depolarization. We're doing our very best to stop the motor loop issues that we see in our kids, but it's hard to tell what the effects we're going to see on an advanced disease in an older patient.
Thanks, Simon. I was thinking more that the mice that have the mutant and the mice that are rescued, is anyone following up the brain volume as well as where it's injected into and where it just, spreads to see whether that volume
I'm not
changes?
sure. Fuck.
That was really kind of as another parameter to look at.
Yeah, it definitely is. And, we've really had to focus our efforts on very, specific things and leave out work that's expensive in other areas. That is one of the things we've left out. But there are papers on the phenotype of this particular model that you can look at, and that do shows kind of brain volume and through MRIs.